Hello students, in the last class we studied tissue system. In the tissue system we studied epidermal as well as vascular tissue system. Now in this class I will be starting with the sectional study of plant organs. Here we will deal with the sectional study of root, stem and leaves. So first of all I will be starting with the sectional study of root in the dicot as well as monocot. Now the outermost covering of any organ represents epidermis. Epidermis, it is made up of thin wall cells which are compactly arranged. So now I am showing outermost layer. Outermost covering, outermost layer of any organ, that is epidermis. Epidermal cells, they are thin, compactly arranged. Compactly arranged as it is root. So over the root epidermis, that waxy layer, what that waxy layer is known as? Cuticle is going to be absent. So it means epidermis is uncutinized. एपिडर्मिस के नीचे प्रेजेंट होगा कॉर्टेक्स. Yes, below the epidermis there is presence of the cortex, that is the part of the ground tissue. Now before starting with the cortex, now these epidermal cells, we know they show tubular outgrowth. The tubular outgrowth which arise from epidermal cells, they represent root hairs. These tubular outgrowth which I have formed here, they represent root hairs, or they are also known as piliferous cells. Piliferous cells. Due to the presence of the piliferous cells, this epidermis, it is also known as piliferous layer. Now, below the epidermis, I will be dealing with the part of the ground tissue. That ground tissue is known as cortex. As we have discussed in the previous class, ground tissue is divided into the three main parts, that is the cortex, pericycle and pith. So first of all, I will be dealing with the cortex here. Epidermis के नीचे जो cells present है, in most of the monocots and few dicots, these cells, they have thickening of suberin. Yes, they have thickening of suberin. These suberized cells, this suberized layer, it is known as exodermis. So I am forming here exodermis. यहाँ पर जो cells हैं, वो thick हैं. और थिकनिंग किसकी है? सुपरिन की. तो ये मैंने बनाई है सुपराइज्ड लेयर. यस, दिस सुपराइज्ड लेयर, इट इज नोन एज एक्सोडर्मिस. This suberized layer, it is present in the most of the monocots and few dicots. Now below this layer, now there is presence of thin cells, parenchymatous cells, and the function of these parenchymatic cells, that is involved in the storage. तो इसके नीचे thin cells, parenchymatous cells, और इन parenchymatous cells का काम है storage of food. तो ये तो हो गया कॉर्टेक्स का आउटर मोस्ट पार्ट, ये हो गया मिडल पार्ट. नाउ द इनर मोस्ट लेयर ऑफ द कॉर्टेक्स. Innermost layer of cortex, it consists of single layer of thick walled cells, and this single layer of thick walled cells represents endodermis, very very important layer. So this is outer region, this is middle region, and now the innermost part of cortex. I am forming it here. Innermost part of the cortex, that is endodermis. सिर्फ single layer, always and always endodermis, represented by single layer of cells. So you can see here the cells of the endodermis, they are swollen. एंडोडर्मिस के सेल आपको फूले हुए नजर आ रहे हैं. The cells are swollen. So you can see the cells of the endodermis, they are barrel shaped. So single layer of cells going to represent endodermis. The cells, they are barrel shaped. Cells, they are thick. The cells, they are thick, and one of the thickening which is present in these cells, that is of suberin. Along with the suberin, there is also presence of lignin. अब ये जो थिकनिंग प्रेजेंट है इन सेल्स के अंदर, दिस थिकनिंग इज प्रेजेंट इन द फॉर्म ऑफ स्ट्रिप्स. यस, दिस थिकनिंग इज प्रेजेंट इन द फॉर्म ऑफ द बैंड्स. थिकनिंग present in the form of strips, present in the form of bands. These bands, they were first discovered by the scientist Casper. सबसे पहले इन बैंड्स को डिस्कवर करा कैस्पर ने. That's why these bands, these strips, they are known as Casparian strips, after the name of the scientist Casper, known as Casparian strips. Now these Casparian strips, they are present in the radial walls, as well as they are also present in tangential walls, right? Let's say this is our endodermal cell of endodermis. This wall and this wall, these walls, they are going to represent radial walls. तो ये दोनों radial walls हो गई. This wall and this wall, this is going to represent tangential wall. अब थिकनिंग कहाँ पर है? रेडियल वॉल्स पे और टेंजेंशियल वॉल में. लेट्स से दिस इज आवर थिकनिंग. यस, दिस इज आवर थिकनिंग. नाउ द थिकनिंग इज प्रेजेंट ऑन द वॉल लाइक दिस. यस, पूरी वॉल के ऊपर थिकनिंग नहीं है, सिर्फ एक बैंड है. ऐसे एक बैंड ऐसे यहां प्रेजेंट है, और एक बैंड नीचे यहां पर प्रेजेंट है. तो पूरी वॉल के ऊपर थिकनिंग नहीं है. द थिकनिंग इज प्रेजेंट इन द फॉर्म ऑफ द बैंड्स, इन द फॉर्म ऑफ द स्ट्रिप्स. एंड व्हाट दी स्ट्रिप्स, दे आर नोन एज कैस्पेरियन स्ट्रिप्स. नाउ एंडोडर्मिस के जो सेल्स है वो थिक है, कैस्पेरियन स्ट्रिप्स प्रेजेंट है, बट सम ऑफ द सेल्स ऑफ द एंडोडर्मिस, दे आर थिन, एंड दीज थिन सेल्स, दे आर नोन एज पैसेज सेल्स. Some cells, they are thin, and these thin cells, they are known as passage cells. Yes, as the name suggests, passage cells, they are involved in the transport of the compounds. They are involved in the transport of water from the outer part of the root towards the inner part. Now, below the endodermis, now the another part of the ground tissue, that is pericycle. एंडोडर्मिस के नीचे अब ये जो मैं लेयर बना रही हूं, दिस इज पेरिसाइकल. Pericycle can be single layered or it can be more than one layer. This one is pericycle. Now, pericycle is very important layer in the root. The pericycle, it will perform some important function. Let's see what are these functions in the case of the dicot and monocot root. In the monocot root, pericycle is involved in the formation of lateral roots. Pericycle का काम है roots बनाना. Which roots? Lateral roots. It is the site of origin of lateral roots. In the case of the dicots, again the function is same. Roots बनाने में help करेगी. But along with that, pericycle in the dicot root is also involved in the formation of cambium. Also involved in the cambium formation. Now, cambium is of two types, vascular cambium and cork cambium. Two type of the cambium, vascular and cork cambium. These two types of the cambium we will discuss in the detail while studying the secondary growth. But you must know here that vascular cambium का सिर्फ कुछ पार्ट बनेगा pericycle की help से and the complete cork cambium is formed with the help of pericycle. Are you getting this? Pericycle will forming the vascular cambium only its part. But complete cork cambium is formed due to the activity of pericycle. तो यहीं पर लिखिए आप vascular cambium का सिर्फ कुछ पार्ट बनेगा and the cork cambium is completely formed by pericycle. Now, after the pericycle, now we will see what is the status with respect to the vascular bundles. As we have discussed that in the root, the types of vascular bundles which are present, they are radial vascular bundle. Radial vascular bundles के अंदर xylem and phloem bundles, they are different and they are present on different radii having alternate arrangement, that is one xylem and another phloem bundle. Are you getting this? There is presence of radial vascular bundle, xylem bundle alag, phloem bundle alag. Along with that, there is presence of exarch condition. Exarch condition, that is the protoxylem that is present towards the outer side. The same condition is there in the case of dicot. अब मोनोकॉट के अंदर जो नंबर ऑफ xylem and phloem vascular bundles है, they are more than 6. Yes, xylem and phloem bundles each, they are more than 6. And this type of condition, it is known as polyarch condition. पर dicots के अंदर जो vascular bundles है, उनका नंबर कम है. The number of the vascular bundles going to range from 2 to 6. So you can call this condition as diarch to hexarch condition. So you're getting this? In the monocots, the number of vascular bundles more than 6, each for xylem as well as phloem. But in the case of the dicot, it is going to be 2 to 6 condition. So let's make the vascular bundles here in this diagram. तो ये मैं बना रही हूं xylem bundles. Yes, these are xylem bundles. So पूरी तरफ आप ऐसे xylem bundles बना दीजिए. Xylem bundles. Yes, these are xylem bundles. So you can see here, these xylem elements, they are smaller in size. The smaller size elements, they are known as protoxylem. These elements, they are larger in size. These elements, they are going to represent metaxylem. तो ये जो छोटे वाले elements हैं, these are protoxylem. ये जो बड़े वाले एलिमेंट्स हैं, these are metaxylem. और प्रोटोजाइलम कहां पर है? बाहर की तरफ. Yes, protoxylem is towards the outside of the organ. This condition is known as Exarch condition. अब प्रोटोजाइलम, मेटाजाइलम के बाद नाउ दी फ्लोएम बंडल्स. एक जाइलम का पैच, एक फ्लोएम का, एक जाइलम का पैच, एक फ्लोएम का. सो आई एम मेकिंग नाउ फ्लोएम बंडल्स. यस, तो हर एक जाइलम बंडल के बाद आप एक एक फ्लोएम का बंडल बना दीजिए. Yes, these are phloem bundles. Now ये xylem bundle है और ये phloem bundle है. इन दोनों के बीच में there is presence of non-vascular tissue. This non-vascular tissue which is present, it is known as conjunctive tissue. Are you getting this? दो बंडल्स के बीच में जो नॉन वैस्कुलर टिश्यू है, दैट टिश्यू इज नोन एज कंजंक्टिव टिश्यू. यस, दिस इज कंजंक्टिव टिश्यू. राइट. Conjunctive tissue is made up of thin wall cells. It is parenchymatous in nature. Yes, made up of parenchyma. Now, in the central part of the organ, there is presence of pith. जो सेंटर वाला पार्ट है, यहां पर pith present है. Pith generally made up of thin cells. These thin cells, they are parenchymatous in nature. अगर मोनोकॉट स्टेम है तो pith is large, it is well developed. But if it is dicot root, अगर मोनोकॉट रूट है तो pith is large, well developed, as you can see here. तो यहां पर तुम देख रहे हो, मैंने यहां पर मोनोकॉट सेक्शन दिखाया है. Why? Because the number of the vascular bundles for phloem as well as xylem, they are more than 6. And in this case, the pith is well developed. पर अगर dicot root होगी, the pith is small or either it is totally absent. So let's write this difference with respect to pith. या तो pith, यहां पर pith large है, or you can say the pith is well developed. In the case of the dicot, the pith either it is small or it is totally absent. So these are the major differences between the monocot and dicot root. So I am quickly recapping this. Outermost layer, epidermis. ये सारा part, ये क्या है? Cortex. 3 layer. This layer, exodermis. Middle layer is parenchymatous, and this thick single layer, that is endodermis. फिर ये जो मैंने बनाया, this is pericycle. These are vascular bundles, and in the center there is presence of pith, which is generally parenchymatous in nature. Now, after looking into the sectional study of the root, let's start with the stem now.